Sir, Gajki sir.
Yes, sir, I'm here. I'm ready.
I'm
I'm ready, sir. Namaste.
Namaste, 1 minute.
Namaste, madam. Very good evening.
Good evening to you, sir.
ಚೆನ್ನಾಗಿದ್ದೀರಾ ಮೇಡಮ್?
ನಾನು ಚೆನ್ನಾಗಿದ್ದೀನಿ, ತಾವು ಹೇಗಿದ್ದೀರಾ?
ಚೆನ್ನಾಗಿದ್ದೀನಿ ಮೇಡಮ್. Thank you very much.
Good, sir. Thank you.
ಇನ್ನೊಂದು 2 minutes madam, we will start session.
No issue, no issue. Yes, sir. Yes.
Yes madam, can we start now? First and foremost, very good evening to all. ಎಲ್ಲರಿಗೂ ಶುಭ ಸಾಯಂಕಾಲ. ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ, ಕರ್ನಾಟಕ ಸರ್ಕಾರ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖೆಯಿಂದ ಹಾಗೂ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಪ್ರೋತ್ಸಾಹಕ ಸೊಸೈಟಿ ಇವರ ಸಂಯೋಜನೆಯೊಂದಿಗೆ ಪ್ರತಿಭಾ ಶೋಧ ಕಾರ್ಯಕ್ರಮವನ್ನು ಹಿಂದಿನ ಸಾಲಿನಿಂದ ನಡೆಸಿಕೊಂಡು ಬರ್ತಾ ಇದೆ. ಈ ಸಾರಿಯು 30 ರಿಂದ 35 ವಿದ್ಯಾರ್ಥಿಗಳನ್ನ ಆಯ್ಕೆ ಮಾಡಿ ಅವರಿಗೆ ಈ ವಿಶೇಷ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ನಡೆಸಲಾಗುತ್ತಾ ಇದೆ. ಅದರ ಜೊತೆಗೆ ಕೆಲವು ಕಾಲೇಜು ಶಿಕ್ಷಕರು ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಸೇರ್ಕೊಂಡು ಈ ಉಪನ್ಯಾಸಗಳ ಲಾಭವನ್ನು ಪಡ್ಕೋಬಹುದು. ಆಮೇಲೆ ಈ ಉಪನ್ಯಾಸಗಳು ಯೂಟ್ಯೂಬ್ ನಲ್ಲೂ ಲೈವ್ ನಡೀತಾ ಇರುತ್ತೆ. ಅಲ್ಲೂ ಕೂಡ ವೀಕ್ಷಕರು ಇದನ್ನ ವೀಕ್ಷಿಸಬಹುದು. ವಿದ್ಯಾರ್ಥಿಗಳು ಹಾಗೂ ಶಿಕ್ಷಕರು, ಯಾವುದೇ ವಿಜ್ಞಾನಾಸಕ್ತರು ಕೂಡ ಈ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದ ಲಾಭವನ್ನು ಪಡೆದುಕೊಳ್ಬಹುದು. ಮೊದಲನೇದಾಗಿ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವ್ರಿಗೆ ನಾನು ತುಂಬಾ ಧನ್ಯವಾದಗಳನ್ನು ಹೇಳಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಯಾಕಂದ್ರೆ ನಮ್ಮ ಈ ಕರೆಗೆ ಹೋಗೊಟ್ಟು ಹಿಂದಿನ ವರ್ಷದಲ್ಲಿ ಉಪನ್ಯಾಸಗಳನ್ನು ನೀಡಿದಂತೆ ಈ ಸಾರಿಯೂ ಕೂಡ ಅವರ ಬ್ಯುಸಿ ಶೆಡ್ಯೂಲ್ ನಲ್ಲೂ ನಮ್ಗೆ ಟೈಮ್ ಅನ್ನು ಕೊಟ್ಬಿಟ್ಟು ಈ ಉಪನ್ಯಾಸಗಳನ್ನು ನೀಡ್ತಾ ಇದ್ದಾರೆ. ನಾವು ಅದಕ್ಕೆ ತುಂಬಾ ಆಭಾರಿಯಾಗಿದ್ದೇವೆ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್ ಮೇಡಮ್ ಫಾರ್ ದಟ್. ಸೊ ಅದೇ ರೀತಿ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿವಿಧ ವಿದ್ಯಾರ್ಥಿಗಳು ಆಯ್ಕೆಯಾದ ಈ ಪ್ರತಿಭಾ ಶೋಧ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಆಯ್ಕೆಯಾದ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ, ಆಮೇಲೆ ಇದರಲ್ಲಿ ಭಾಗವಹಿಸುತ್ತಿರುವ ವಿವಿಧ ಕಾಲೇಜುಗಳ ಶಿಕ್ಷಕರಾಗಿರ್ಲಿ, ಹಾಗೆ ಇನ್ನಿತರ ವಿಜ್ಞಾನಾಸಕ್ತರು ವೆಬೆಕ್ಸ್ ಸಿಸ್ಕೋ ಪ್ಲಾಟ್ಫಾರ್ಮ್ ಅಲ್ಲಿ ಹಾಗೂ ಯೂಟ್ಯೂಬ್ ನಲ್ಲಿ ನಮಗೆ ಈ ಇದರಲ್ಲಿ ಉಪನ್ಯಾಸವನ್ನು ಸೇರಿಕೊಂಡಿರೋರಿಗೆ ಎಲ್ಲರಿಗೂ ಸುಸ್ವಾಗತ ಹೇಳಿಸ್ತಾ, ಈ ಉಪನ್ಯಾಸ ಪ್ರಾರಂಭ ಮಾಡಕ್ ಮುಂಚೆ ನಾನು ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವ್ರ ಬಗ್ಗೆ ಒಂದೆರಡು ಮಾತು ಹೇಳಕ್ ಶುರು ಮಾಡಿ ಅವರ ಪ್ರೊಫೈಲ್ ಬಗ್ಗೆ ಹೇಳ್ತೀನಿ. Dr. Lakshmi Murthy is a senior biology teacher and she is a trainer and adolescent counselor with over 25 years of experience. Currently, she heads the program. She is a program head of Medical Excel at Base Educational Service Private Limited and HOD of Biology since 2018. In addition, she is a senior biology faculty at Base Educational Services Private Limited since 2016. And her educational qualifications are MS in Botany, B.Ed certificate in counseling skills. She has many awards and recognitions. She has received personal commendation letters from Ministry of HRD, Government of India, for her contribution in the education sector. And she has received Best Biology Teacher Award from the Indian Brain Bee organization. Also, she received a Reliance Foundation Award for her contribution to the biology teaching, and she has secured the first rank for senior school biology in the Teachers Professional Olympiad, which is well known as TPO for 2018 and 2019 at all India and UAE level. Congratulations to you, madam. On behalf of all the participants and on behalf of KST, we congratulate you for this. And apart from this, she has many work experience in educational institutes. She taught middle school chemistry and biology at Phoenix School, Belgium, and taught biology for secondary school and senior school as a senior faculty at NPS, National Public School, Indiranagar and Rajajinagar from 2000 to October 2015. She was senior school student counselor at National Public School, Rajajinagar, and she was head and senior coordinator and also a guide for adolescent education program at NPS Rajajinagar. She was the head and senior coordinator for the drug abuse awareness program at National Public School, Rajajinagar. Also, head and senior coordinator for the continuous and comprehensive education program at the National Public Schools. She was the teacher trainer for the CCE, AEP, and life skills program at the National Public Schools. Apart from this, she trained in dramatic and Carnatic vocalist, and she enjoys reading and artwork. With this brief introduction, I welcome to you this talent search program webinar, ma'am. Uh, before beginning the session, I request students to listen the session carefully, and as we requested, after the question we declare from madam, you have to write it in a paper and mention the details and scan it and send it to the mentioned email ID. Thank you. With this, handing over mic to madam. The platform is yours, ma'am. You can continue. Thank you very much.
Thank you so much for that introduction, really humble, sir. And I'm so glad to be a part of KSTA. And it's always a pleasure to address students and speak about my favorite subject, which is biology. And a lot of students, they really like to learn more about biology. They can relate to it very easily. So, ಈಗ ಒಂದು life ಅಂತ ಅಂದ್ರೆ ಏನು ಅಂತ ಕೇಳಿದ್ರೆ, do we need a heart to show that life exists? Do we need a brain to show that life exists? ಒಂದು blood vessel ಬೇಕಾ, ಒಂದು nerve ಬೇಕಾ ಅಂತ ಕ್ವೆಶ್ಚನ್ ಬರುತ್ತೆ. ಅವೆಲ್ಲ ಏನೂ ಬೇಡ. ಲೈಫ್ ಅಂತ ತೋರಿಸಕ್ಕೆ ಎರಡೇ ಸಾಮಾನು ಬೇಕು. One is cytoplasm and the other is cell membrane. So our introduction in that, let us start with one of the most important components that shows that life exists, and that is going to be cell membrane. Makkale niಮಗೆ ಗೊತ್ತಾ, past 40-50 ವರ್ಷದಿಂದ most research that has happened in the field of biology has happened on the cell membrane. Whether you want to learn how a particular drug, ನೀವು ಡೋಲೋ 650 ಕೇಳಿರ್ಬೋದು, ಆ ಡ್ರಗ್ ನ ಒಂದು ಪರ್ಸನ್ ತಗೊಂಡ್ರು ಯಾಕೆಂದ್ರೆ ಅವ್ರಿಗೆ ಫೀವರ್ ಇದೆ ಅಂದ್ರೆ ಅದು ಹೇಗೆ ಆ ಫೀವರ್ ನ ಕಡಿಮೆ ಮಾಡುತ್ತೆ ಅಂದ್ರೆ ಮತ್ತೆ ವಾಪಸ್ ನಾವು ಬರುತ್ತೆ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ಗೆ. ಒಂದು ಪರ್ಸನ್ಗೆ ಲೈಫ್ ಸ್ಟೈಲ್ resistant diabetes ಅಂತ ಬರುತ್ತೆ. ಲೈಫ್ ಸ್ಟೈಲ್ ಇಂಡ್ಯೂಸ್ಡ್ ಇನ್ಸುಲಿನ್ ರೆಸಿಸ್ಟೆಂಟ್ ಡಯಾಬಿಟೀಸ್ ಅಂದ್ರೆ ಟೈಪ್ 2 ಡಯಾಬಿಟೀಸ್. ಅವ್ರಿಗೆ ಇನ್ಸುಲಿನ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ. ಅದರ ಪ್ರಾಬ್ಲಮ್ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಸೆಲ್ಸ್ ಆರ್ ಅನೇಬಲ್ ಟು ರೆಕಗ್ನೈಸ್ ಇನ್ಸುಲಿನ್. ಅದಕ್ಕೋಸ್ಕರನೇ ಡಯಾಬಿಟೀಸ್ ಮೆಲಿಟಸ್ ಬರುತ್ತೆ ಅವ್ರಿಗೆ. ಸೊ ಹಂಗಾಗಿ ಏನಾಗುತ್ತೆ, ಈವನ್ ದೋ ದೇರ್ ಇಸ್ ಇನ್ಸುಲಿನ್, they will have all signs and symptoms of diabetes mellitus. ಇದು ಯಾವ್ದ್ರಿಂದ ಆಗೋದು? Again, the issue is going to be with respect to cell membrane. So ಹಾಗಾಗಿ cell membrane relevance is going to be much more than what we think. And let us learn about cell membrane. ಅದು ಏನು functions ನ ಮಾಡುತ್ತೆ ಅಂತ ತಿಳ್ಕೊಳ್ಳೋಕ್ಕಿಂತ ಮುಂಚೆ, let us try and learn the structure of cell membrane. Cell membrane ಏನು structure ಇರಬಹುದು ಅಂತ, the history is going to go back right up to the 19th century. 19th century, we had a number of biochemists and a number of cell biologists who were very much interested to find out what is the structure of a cell? As you are aware, Robert Hooke discovered the cell and also gave the name cell, but what he observed was just dead cell. And he thought that there is nothing present within a cell. But as years passed, there were a number of cases where biologists were able to take live tissue, fruit piece ಆಗಿರಬಹುದು, leaf piece ಆಗಿರಬಹುದು, a drop of blood ಆಗಿರಬಹುದು. They started observing more and more cells, and they realized that cell is not an empty cell wall like what Robert Hooke had seen. Instead, cell has got a lot of interesting components, and one of the components which is present in all the 5 kingdoms. Tamge gotta yavu 5 kingdoms living organisms alli andre, one kingdom is monera, innondu protista, innondu fungi, innondu plante, amele animalia. All 5 kingdoms alli you find that living cells will require this structure to be present called as cell membrane. Apart from this, you may find organisms having additional boundaries present. For example, fungal cell andre cell membrane jotege avage kaditende cell wall irutte. Or if it is a bacterial cell, along with cell membrane, it may show the presence of a cell wall made up of peptidoglycan and glycocalyx layer will also be present. So, cell membrane is by default seen for all living cells of the 5 kingdoms. Now, this cell membrane story, as I said, started in the 19th century. Lot of different scientists put forth their own versions about what is the cell membrane. Before we go to the structure and functions of the cell membrane, ಮಕ್ಕಳಿಗೆ ಯಾವತ್ತು ಈಚೆಗೆ ನಾವು ಚಿಕ್ಕವರಾದಾಗ ಅಲ್ಲ, ಮಕ್ಕಳು ಏನ್ ಕೇಳ್ತಾರೆ, ಇದು ನಾನು ಕಲ್ತ್ಕೊಂಡ್ರೆ ನನಗೆ ಏನು ಬೆನಿಫಿಟ್? Many of you all are going to become scientists, and who knows, you may find wonderful things about how cell membrane behaves. Some of you may want to become engineers. Engineering exams, ಅದು national level JEE mains exam ಆಗಿರಬಹುದು, JEE advanced exam ಆಗಿರಬಹುದು, ಇಲ್ಲ ನಮ್ಮ local KCET exam, government exam, ಅದರ ಯಾವುದೇ exam ಆಗಿದ್ರುವೆ, even though you would want to write a entrance exam paper in PCM, physics, chemistry, maths ಅಲ್ಲಿ ಬರಿಬೇಕಾದ್ರುವೆ, please understand, dear students, that cell membrane is just a bunch of chemicals, and a chemistry question can also appear on the cell membrane. So what I'm going to teach now is definitely relevant to all the students, irrespective of what you want to do later. What about students who want to take pure science? Iga neevu kelirbodu KVPY anta, Kishore Vaidyanik Protsahan Yojana, Government of India ಈ ತರ ಸ್ಕಾಲರ್ಶಿಪ್ ಪ್ರೋಗ್ರಾಮ್ ಮಾಡಿದೆ. ಬಹಳ ಜನ ಮಕ್ಕಳು KVPY ಎಕ್ಸಾಮ್ಸ್ ಬರೀತಾರೆ. ಫಸ್ಟ್ ಇಯರ್ PUCB, ಸೆಕೆಂಡ್ ಇಯರ್ PUCB. ಆ ಪರೀಕ್ಷೆಯಲ್ಲೂ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಆಧಾರಿತ ಕ್ವೆಶ್ಚನ್ಸ್ ಬಹಳ ಬರುತ್ತೆ. So let us not delay at all. With that brief introduction, let us immediately go to cell membrane. So as I said, cell membrane, what is the structure alone? Lot of scientists had done their own studies. We had a scientist group team called as Danieli and Davison. They gave their own version to what a cell membrane is going to be. We had scientists called as Robertson, who gave his own version about what a cell membrane is going to be. But none of the scientists, other scientists, were convinced with respect to this structure. Why was there a problem? Was there a problem with Danieli and Davison's model? Was there a problem with Robertson's model? No, the model had a problem because it only explained the structure of a cell membrane. I'm sure as students of science, you will appreciate ನಾವು ಯಾವುದೇ ಒಂದು ಮಾಡೆಲ್ ನ ಪ್ರಪೋಸ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಅದು ಸ್ಟ್ರಕ್ಚರಲ್ ಆಸ್ಪೆಕ್ಟ್ಸ್ ಆಸ್ ವೆಲ್ as ಫಂಕ್ಷನಲ್ ಆಸ್ಪೆಕ್ಟ್ಸ್ ನ ಖಂಡಿತ ಮೀಟ್ ಮಾಡಬೇಕು. ನಾವು ಒಂದು ಕಾರ್ ನ ಡಿಸೈನ್ ಮಾಡಬೇಕು ಅಂದ್ರೂ ಅದು ಹೇಗೆ ಜನರನ್ನ ಕುತ್ಕೊಳ್ಳಕ್ಕೆ ಒಂದು ಅರೇಂಜ್ಮೆಂಟ್ ಮಾಡುತ್ತೋ ಅದೇ ತರ ಹೇಗೆ ಅದು ನೀಟ್ ಆಗಿ ಏರ್ ನ ಕಟ್ ಮಾಡ್ಕೊಂಡು ಮುಂದೆ ಹೋಗುತ್ತೆ ಅನ್ನೋ ತರ ಬೋತ್ ಫಂಕ್ಷನಲ್ ಸ್ಟ್ರಕ್ಚರಲ್ ನಾನು ಒಂದು ಮಾಡೆಲ್ ನ ಪ್ರಪೋಸ್ ಮಾಡ್ಲೇಬೇಕಾಗುತ್ತೆ. Previous scientists who had proposed this model, they failed to give the explanation as to how cell membrane shows a property called selective permeability. Hagagi, in the year 1972, we are going to come across two scientists who are Seymour Singer, ಮುಂದೆ ನಿಮಗೆ ಅವರು ಪಿಕ್ಚರ್ಸ್ ತೋರಿಸ್ತೀನಿ ನಾನು. Seymour Singer and Garth Nicholson. These two cell biologists, they analyzed the previous scientists' work. They also did tremendous amount of work on human RBC cell membrane, and by using the any kind of data that they got from the previous scientists' data, as well as by analyzing human RBC cell membrane, they were able to put forth a model to explain both the structure and function of cell membrane. And that particular model, which has found wide acceptance from the past 50 years, is called as the fluid mosaic model of cell membrane. And today I am going to be explaining the fluid mosaic model of cell membrane to you. Before I go to the functions, let us look at the structure that Singer and Nicholson have proposed. I'm sure students will be interested to know. Human RBC will matra yaki itara research aitu. Even before Singer and Nicholson, lot of scientists had done cell membrane work on human RBC. Why not take leaf tissue from some plants? Why not take fruit pieces? Why not take meat pieces from chicken to analyze cell membrane? Well, the most important aspect here is when you are trying to analyze the cell membrane, you need a standard cell. And students, I'm sure you would have heard in school level also, human RBCs are perfectly circular in outline, and they have a 7-micron diameter to the cell. Apart from that, we know that they are going to be biconcave disc-shaped structures. So, the surface area of a human RBC is well calculated. ನಾವು ಯಾವುದೇ ಒಂದು ಸರ್ಫೇಸ್ ಏರಿಯಾನ ತಗೊಂಡು ಅದರಷ್ಟೇ ಮಟ್ಟಕ್ಕೆ ಕೆಮಿಕಲ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇದೆ ಅಂದ್ರೆ ಐ ವಿಲ್ ನೋ ದಟ್ ಇಸ್ ಒನ್ ಕೆಮಿಕಲ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಲೆಟ್ ಅಸ್ ಸೇ ನನ್ನ ಕೈಯಲ್ಲಿ ಡಸ್ಟರ್ ಇದೆ. ಈ ಡಸ್ಟರ್ ಸರ್ಫೇಸ್ ಏರಿಯಾ ಎಷ್ಟು ಅಂತ ತಿಳ್ಕೊಳ್ಳಕ್ಕೆ if I wrap it with paper neatly so that there is no overlapping, and then I open the paper and measure the paper size, you will get to know what is the surface area of this duster. Suppose when you measure the surface area, you realize that the surface area appears to be twice paper size. Surface area ginta double bantu andre takshana you will know there is some component of the cell membrane which is present 2 times the surface area. Hangagi standard sizes, as students of physics and maths you will appreciate, standard sizes are very important if you have to mathematically calculate. And human RBCs, unlike parenchyma in plants or maybe muscle cells in a meat piece, they are of standard sizes. So it becomes so easy to study cell membrane structure from human RBCs. Therefore, the preference was with respect to human RBCs itself. Now, going ahead, let's now go through the fluid mosaic model of the cell membrane. In the cell membrane, it may be from a rabbit, It may be from a bacterium, it may be from humans itself. What are the common chemicals present in a cell membrane? First, outline list ಮಾಡ್ಕೊಳ್ಳೋಣ. Many a times, ನಿಮ್ಮ CET question ಇರ್ಬೋದು. Many of you all want to go for yoga and naturopathy through CET. Some of you all want to become doctors for the NEET exam ತಗೊಂಡು ಇಲ್ಲ JEE mains ನಡೆಯದಲ್ಲಿ ಅವಾಗ they will ask you a question, multiple choice question ಕೇಳ್ತಾರೆ. ಇದರಲ್ಲಿ ಯಾವ ಕಾಂಪೊನೆಂಟ್ಸ್ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಅಲ್ಲಿ ಇದೆ ಇಲ್ಲ ಯಾವುದು ಇಲ್ಲ ಅಂತೆಲ್ಲ ಕೇಳ್ತಾರೆ. ಅವಾಗ you must boldly know ನನಗೆ ಗೊತ್ತು ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಅಲ್ಲಿ ಏನೇನಿದೆ ಅಂತ. Definitely you will be in a position to answer it very well. So what are the components in a cell membrane? Cell membrane ಅಲ್ಲಿ you will find lipids are going to be present in a cell membrane. Okay, lipids ide ante yav tara lipids ide anta next question barutte. Ye lipids ide anta. So the lipids which are present in a cell membrane are going to be of two types. Yav types. The first type of lipid and the one which is present in abundance is going to be called as phospholipids. So the one that is present in abundance is called as phospholipids. Phospholipids ಏನು ಅಂತ brief ಆಗಿ ಹೇಳ್ತೀನಿ. I'm sure your chemistry teachers would have also brought about this topic to you. Sarina, second type of lipid that is present is called as cholesterol. How do some of the plants instead of cholesterol they may have only sterols? Definitely irutte, but some kind of a sterol, either modified sterol like cholesterol or natural sterols may be seen. So ಎರಡು ನಾನು ತಗೋತೀನಿ. One is phospholipids and the second is going to be cholesterol. Before we go to the second component, ಇದ್ರ ಬಗ್ಗೆ brief ಆಗಿ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. So cholesterol ನೀವು ನೋಡಿದ್ರೆ I'm sure many of you all will have a question. Ma'am, ನಾನು bad cholesterol ಬಗ್ಗೆ ಕೇಳಿದ್ದೀನಿ. ಅದು heart diseases cause ಮಾಡುತ್ತೆ, artery diseases cause ಮಾಡುತ್ತೆ. That is a different thing. ಅದು ನಿಮ್ಮ diet ಇಂದಾಗಿ ಇಲ್ಲ for some kind of oxidation reasons ಆಗಲಿ you will have some bad cholesterol called as LDL, low density lipoproteins ಅಂತ ಹೇಳ್ತೀವಿ. That may clog arteries. But very cholesterol. Let us not worry about that. Apart from that, good cholesterol is a HDL, high density lipoproteins. So that is good cholesterol, which is required for you to make certain hormones in your body, like some of the adrenal cortex hormones, gonadal hormones. Apart from that, we are looking at cholesterol, which is present as part of the cell membrane. Yellow bandu cell ಒಳಗಡೆ use as a raw material ನೋಡ್ತಾ ಇದೆ. We are speaking about cholesterol. ಈ cholesterol ನ ತಗೊಂಡು ನಾನು steroidal hormones ಮಾಡಬಹುದಾ? ಖಂಡಿತ ಮಾಡಬಹುದು. We are not saying no at all. But this is cholesterol which is part of the cell membrane, right? So cholesterol ಜೊತೆಗೆ ಏನಿರುತ್ತೆ phospholipids ಅಂತ ಹೇಳ್ತೇವೆ. Phospholipids very briefly I will explain to you. So phospholipid ಮಾಡೋಕೆ ನಮಗೆ ಏನೇನು ಬೇಕಾಗುತ್ತೆ? No problem, we will try to find out. When we phospholipid create molecule, you require a molecule called as trihydroxy alcohol. That is glycerol. Okay. Apart from that, you require two fatty acid molecules. Fatty acid molecules, two molecules you need. Saturated irbodu, unsaturated irbodu. That depends on where the organism is going to live. Polar bear ಗೆ ನೋಡಿದ್ರೆ ನಿಮಗೆ unsaturated fatty acids ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಅದೇ ಇಲ್ಲಿ ನಮ್ಮ ಬೆಳ್ಳಾರಿ ಕಡೆ bears ನೋಡಿದ್ರೆ ಕರಡಿಗಳು ನೋಡಿದ್ರೆ ಅಲ್ಲಿ ನಿಮಗೆ ಸ್ವಲ್ಪ saturated fatty acids ಇದ್ರೂ ಪರವಾಗಿಲ್ಲ. So you will have a little bit of difference with respect to what are the components, but it's okay. So two molecules of fatty acids plus ನನಗೆ phosphoric acid. Okay, ನೀವು ಅದನ್ನ phosphate group ಅಂತಾನೂ ಕರೀಬಹುದು. Doesn't matter. Phosphoric acid ಬೇಕು. ಈಗ ಇವನ್ ಮೂರನ್ನು ನೋಡಿದ್ರೆ what you will have ಇಲ್ಲಿ ಒಂದು glycerol molecule ಕೂತಿರುತ್ತೆ. Okay, I will draw it in a diagram. ಈ molecular formula ಹೋದ್ರೆ you will not be able to cover the main topic. So ಇವಾಗ glycerol I will show it in this manner. ಇದು glycerol ಅಂತ ಅನ್ಕೊಳ್ಳಿ. which is component number 1. You will have this joining. This is going to be one fatty acid molecule. This is going to be another fatty acid molecule which is present in this manner. The component is going to be our phosphoric acid. More components irukke. This part of the phospholipid molecule, glycerol, then this is fatty acid. Fatty acids phosphoric acid. This whole part is called as a hydrophilic part of the phospholipid molecule. Hydrophilic and hydro referring to water. Philia and love. So it is okay or it likes water, right? Even chains, fatty acid chains, which has got esterified along with the glycerol molecule, which has formed bonds with the glycerol molecule. Hydrophobic part, because that will be hydrocarbon chains. Carboxylic acid group. So these are hydrocarbon chains. They don't like water. We call them as the hydrophobic part. When we draw a cell membrane, it's a complicated structure. We do not have to draw. Instead, we will be representing henceforth when the phospholipid molecule will be represented in a very let us say simple diagrammatic manner. Phospholipid nano, even in a class, I will just show it in this manner. The hydrophilic head, which is glycerol with the phosphate group. And these two chains here, hydrophobic tail, which is going to be made up of the long hydrocarbon chains of fatty acid. This is a phospholipid molecule. Okay, what about cholesterol? Cholesterol, now, for the sake of diagram, I am going to represent cholesterol simply as a rectangle for today's class. Even in a class, we have cholesterol now, just as a rectangle represent marketing. So that is going to be your component number 1. Okay, component number 2 lipids first component so that we keep that in mind and don't forget about lipids. Either the component second component second and also a major component. Lipids were a major component. Second component, which is definitely a major component, is going to be proteins. I am sure you have heard about this particular biomolecule, proteins. When the polypeptide chain fold proteins, right? And polypeptide chain, of course, you all will know it is going to be a polymer of amino acids. Many amino acids peptide bond join one protein. Today, I will be drawing proteins by using yellow color. Proteins so, I will be drawing proteins today by using yellow color. Draw marta, I will explain to you what are the various types of proteins that are going to be seen. Okay, so remember, second component is going to be proteins. Okay, what is the third component? Third component which is going to be present is called as oligosaccharide. Biology, oligo ಅಂದರೆ small ಅಂತ. Oligo ಅಂದರೆ small. ನೀವು oligo ಕೀಟ ಈ ತರ ಎಲ್ಲ ವರ್ಡ್ಸ್ ಓದಿರಬಹುದು. Okay, so oligonucleotides ಈ ತರ ಎಲ್ಲ ಓದಿರಬಹುದು. So this is going to be oligosaccharide, right? Small, it is like a tiniest polysaccharide. That will be more than sufficient now. So it is a small polysaccharide. So oligosaccharides, which are basically carbohydrates, are also present in your cell membrane. Lipids, proteins, oligosaccharides, that depends on the cell, depends on the organism. For example, if you take human RBCs, Generally, ನಿಮ್ಮ NCERT book ಅಲ್ಲಿ ಬರ್ತಿರೋ ಪ್ರಕಾರ, you have 52% of proteins present in human RBCs and you have around 40% proteins present and the remaining is going to be made up of oligosaccharides ಅಂತ ನಿಮ್ಮ NCERT book ಅಲ್ಲಿ ಬರ್ತಿದೆ. ಅದೇ ತರ if you take some of the neurons, nerve cells ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ, ಅಲ್ಲಿ you will find that you have almost 70 to 85% lipids and just around 15 to 30% proteins in there. So in the same body, nimge lipid and protein proportion will definitely vary, and I'm sure students will find it very interesting. Do you know that there are certain animals, ನೀವು ಓದಿರ್ಬೋದು, ನಮ್ಮ ಕೊಕ್ಕರೆ ಬೇಳೂರು, ಓಕೆ, ಅಲ್ಲಿ ವಲಸೆ ಹಕ್ಕಿಗಳು ಬರುತ್ತೆ. ಬೇರೆ ಬೇರೆ ಏಷ್ಯನ್ ಕಂಟ್ರೀಸ್, ಯುರೋಪಿಯನ್ ಕಂಟ್ರೀಸ್ ಇಂದ ವಲಸೆ ಹಕ್ಕಿಗಳೆಲ್ಲ ಬರುತ್ತೆ, ಮೈಗ್ರೇಟರಿ ಬರ್ಡ್ಸ್. ಓಕೆ, ನೀವು ರಾಜಸ್ಥಾನ್ ಹೋದ್ರೆ ಖರಗ್ಪುರ್ ಸ್ಯಾಂಕ್ಚುರಿ ಸೈಬೀರಿಯನ್ ಕ್ರೇನ್ಸ್ ಎಲ್ಲ ಬರುತ್ತೆ. ದೋಸ್ ಬರ್ಡ್ಸ್ ಸೈಬೀರಿಯಲ್ ಇದ್ದಾಗ ಅದೇ ಸೆಲ್ ಪ್ರಪೋರ್ಷನ್ ಬೇರೆ ಇರುತ್ತೆ. ಇಲ್ಲಿ ನಮ್ಮ ಇಂಡಿಯಾ ಬರೋ ಹೊತ್ತಿಗೆ proportion of lipids and proteins, they are able to change. Very few animals have this ability. Okay, ಅದಕ್ಕೆ homo-viscous adaptation ಅಂತ ಹೇಳ್ತೀವಿ. Homo-viscous adaptation ಅಂದ್ರೆ the ability to change the nature of, or the proportion rather, of lipids and proteins. Even the nature of lipids they can change to suit warm weather and cold weather, etc. Very interesting, no? ಎಷ್ಟು ಚೆನ್ನಾಗಿದೆ ಇದನ್ನೆಲ್ಲ ತಿಳ್ಕೊಳ್ಳೋಕೆ. So let us go ahead and now ಇವೆಲ್ಲ ಮೂರು ಸೇರಿಸ್ಬಿಟ್ರೆ ಎಲ್ಲವನ್ನು ತಗೊಂಡು ಒಂದು ಬೀಕರ್ ಅಲ್ಲಿ ಒಂಚೂರು ಲಿಪಿಡ್ಸ್, ಒಂಚೂರು ಪ್ರೋಟೀನ್ಸ್, ಒಂಚೂರು ಆಲಿಗೋಸ್ಯಾಕರೈಡ್ಸ್ ಹಾಕ್ಬಿಟ್ಟು ಕಲಕ್ಬಿಟ್ರೆ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ಸ್ ಸಿಗತ್ತಾ? ಖಂಡಿತ ಇಲ್ಲ. ವಾಟ್ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಇಸ್ ಹೌ ಆರ್ ವಿ ಗೋಯಿಂಗ್ ಟು ಬಿ ಅರೇಂಜ್. ಅರೇಂಜ್ಮೆಂಟ್ ಬಹಳ ಮುಖ್ಯ, ಯಾವುದೇ ಆಗಿರಲಿ, ಸರಿನಾ? ಲೆಟ್ ಅಸ್ ಗೋ ಅಹೆಡ್ ಅಂಡ್ ಲುಕ್ ಎಟ್ ದಿ ಅರೇಂಜ್ಮೆಂಟ್ ಆಫ್ ದೀಸ್ ತ್ರೀ ಕಾಂಪೋನೆಂಟ್ಸ್ ಇನ್ಸೈಡ್ ದ ಸೆಲ್ ಮೆಂಬ್ರೇನ್. So first ಏನಾಗುತ್ತೆ ನಿಮಗೆ, ನೀವು ಯಾವುದೇ ಆರ್ಗ್ಯಾನಿಸಮ್ ತಗೊಳ್ಳಿ, ನಾವು ದೊಡ್ಡ ವರ್ಡ್ಸ್ ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಸ್ ಟೆರೆಸ್ಟ್ರಿಯಲ್, ದಿಸ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಸ್ ಅಕ್ವಾಟಿಕ್, ಏನೇನೋ ಹೇಳ್ತೀವಿ, ಕರೆಕ್ಟ್, ತಪ್ಪಲ್ಲ. ಬಟ್ ವೆನ್ ಯು ಲುಕ್ ಅಟ್ ಎನಿ ಸೆಲ್, ಲಿವಿಂಗ್ ಸೆಲ್ ತಗೊಳ್ಳೋಣ, ಡೆಡ್ ಸೆಲ್ ಬಗ್ಗೆ ಮಾತಾಡೋಣ. ಎನಿ ಲಿವಿಂಗ್ ಸೆಲ್ ತಗೊಂಡ್ರೆ, ವೆದರ್ ಇಟ್ ಇಸ್ ಅ ಟೆರೆಸ್ಟ್ರಿಯಲ್ ಆನಿಮಲ್ ಆರ್ ಅನ್ ಅಕ್ವಾಟಿಕ್ ಆನಿಮಲ್, ದ ಎನ್ವೈರನ್ಮೆಂಟ್ ಫಾರ್ ಎವ್ರಿ ಸೆಲ್ ಇಸ್ ಸ್ಟಿಲ್ ಅಕ್ವಾಟಿಕ್. ಅದರದ್ದು ಹೊರಗಡೆ ಒಂದು ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಹೊರಗಡೆ ಆಯ್ತು, ಒಳಗಡೆ ಆಯ್ತು, ಆಕ್ವಿಯಸ್ ಎನ್ವೈರನ್ಮೆಂಟೇ ಇದೆ. ಅದನ್ನ ಲೈಫ್ ಹ್ಯಾಸ್ ಬೀನ್ ಏಬಲ್ ಟು ಗಿವ್ ಅಪ್ ಟಿಲ್ ನೌ. ಅದನ್ನ ಒಂದನ್ನ ನಾವು ಕಾಯ್ತೀವಿ. ಸೊ ಫಾರ್ ಎವ್ರಿ ಸೆಲ್ ದಿ ಎನ್ವೈರನ್ಮೆಂಟ್ ಇಸ್ ಸ್ಟಿಲ್ ಅಕ್ವಾಟಿಕ್. ಆ ವರ್ಡ್ ಯೂಸ್ ಮಾಡದೇ ಇರೋದು ಆಕ್ವಿಯಸ್ ಎನ್ವೈರನ್ಮೆಂಟ್ ಅಂತ ಹೇಳ್ಬೋದು. ಆರ್ ಇನ್ ಅದರ್ ವರ್ಡ್ಸ್, ಒಂದು ಅಮೀಬಾ ಇರುತ್ತೆ, ಐ ಆಮ್ ಶೋರ್ ನೀವು ಅಮೀಬಾ ಬಗ್ಗೆ ಕೇಳಿದ್ದೀರಾ. ಸೊ ಒಂದು ಅಮೀಬಾ ಸೆಲ್ ಮೆಂಬ್ರೇನ್, ಲೆಟ್ ಅಸ್ ಸೇ ಒಂದು ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ನ ತಗೊಂಡು ಅದನ್ನ ಬಹಳ ಮ್ಯಾಗ್ನಿಫೈ ಮಾಡಿದ್ರೆ, ಆಫ್ ಕೋರ್ಸ್ even electron microscope that gives you highest level of magnification, ಆತರ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲೂ you will not find cell membrane structure ನಾ ಹೇಳಿದಂತೆ ಕಾಣಿಸಲ್ಲ. Okay, because we are speaking to molecular level, but definitely we will be able to see cell membrane in a little bit more detail. ನಿಮಗೆ ಕಂಪೌಂಡ್ ಮೈಕ್ರೋಸ್ಕೋಪ್, that is what is called as light microscopy, ಅದರಲ್ಲಿ ಅಂತ ಡೀಟೇಲ್ ಸ್ಟ್ರಕ್ಚರ್ ಸಿಗಲ್ಲ. So when we take an amoeba at the cell membrane ಹೇಗೆ ಅಂತ ಹೇಳ್ಕೊಂಡ್ರೆ, this is how the cell membrane is going to be. Remember, amoeba ಗೆ cytoplasm ಇದೆ aqueous based and ಹೊರಗಡೆ water ಇದೆ aqueous based. ನಿಮ್ಮ RBC ತಗೊಳ್ಳಿ, ಅದ್ರ ಒಳಗಡೆ cytoplasm aqueous based, ಹೊರಗಡೆ ಇರೋ ಪ್ಲಾಸ್ಮಾ ಬ್ಲಡ್ aqueous based. That is true for all the living cells in the living world. So ಹಂಗಿರುವಾಗ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ, ಈ ಕಡೆಯೂ ನೀರು, ಈ ಕಡೆಯೂ ನೀರು. ಓಕೆ, ಹಾಗಾಗಿ ಆ ಪಾರ್ಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಆಕ್ಯುಪೈಡ್ ಬೈ ದ ಹೈಡ್ರೋಫಿಲಿಕ್ ಹೆಡ್ಸ್ ಆಫ್ ದ ಫಾಸ್ಫೋಲಿಪಿಡ್ಸ್. ಎರಡೂ ಕಡೆ they will get assembled. How will they get assembled? This is how the assembly is going to be. So I'm going to have here, this is a phospholipid family, right? The phospholipid. So I'm drawing some phospholipids for your reference here. It is just diagrammatic representation. Please remember, Sarina, in electron microscope, it is details, but just ಮಾಡಲ್ಲ. So when you look at this, you will find ಇಲ್ಲೆಲ್ಲ ನಿಮ್ಮ ಹೆಡ್ಸು this is going to be the environment side. ಇದು environment side. ಇಲ್ಲಿ ಬರೋದು ನಿಮ್ದು ಸೈಟೋಸೋಲ್ ಸೈಡ್ ಅಂದ್ರೆ ಸೈಟೋಪ್ಲಾಸ್ಮಿಕ್ ಸೈಡ್ ಇರುತ್ತೆ. ಹಾಗಾಗಿ ಎರಡು ಆಕ್ಟಿವಿಸ್ ಇರೋದ್ರಿಂದ the lipids get arranged in this manner. ಸರಿನಾ? ಈ ತರ ಅರೇಂಜ್ಮೆಂಟ್ ನಿಮ್ದು ಲಿಪಿಡ್ಸ್ ಬಂದ್ಬಿಡುತ್ತೆ. ನಾನು ಇದೇನಿದು ಒಂದು ಗ್ಯಾಪ್ ಬಿಟ್ಬಿಟ್ಟಲ್ಲ, ಡೋಂಟ್ ವರಿ, ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಫಿಲ್ ಅಪ್ ದಟ್ ಗ್ಯಾಪ್ ಬಿಕಾಸ್ ಲಿಪಿಡ್ಸ್ ಮಾತ್ರ ಇರಲ್ಲ. ಯಾವುದೇ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಅಲ್ಲಿ ಮೂರು ಕಾಂಪೊನೆಂಟ್ಸ್ ಲಿಪಿಡ್ಸ್, ಪ್ರೋಟೀನ್ಸ್, ಆಲಿಗೋಸ್ಯಾಕರೈಡ್ಸ್ ಮೂರನ್ನು ಅಕಾಮಿಡೇಟ್ ಮಾಡಿದ್ರೆ ಐ ಆಮ್ ಟ್ರೈಂಗ್ ಟು ಡ್ರಾ ದ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಇನ್ ದಿಸ್ ಮ್ಯಾನರ್. ಇವಾಗ ಈ ತರ ನಾನು ಒಂದು ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಓಕೆ ಒಂದಷ್ಟು ನಾವು ಫಾಸ್ಫೋಲಿಪಿಡ್ಸ್ ಡ್ರಾ ಮಾಡಿದ್ವಿ. I'm sure as students of chemistry you learn ಆಗುತ್ತೆ. Suppose you take ಒಂದು oil ನ ತಗೊಂಡ್ಬಿಟ್ಟು ನೀವು ಒಂದು ಗ್ಲಾಸ್ ವಾಟರ್ ಮೇಲೆ ಹಾಕಿದ್ರೆ ಮೇಲ್ಗಡೆ ಆಯಿಲ್ ಫ್ಲೋಟ್ ಆಗುತ್ತೆ. ಯಾಕೆ? ಆ ಮಾಲಿಕ್ಯೂಲ್ಸ್ have got aligned such that the hydrophilic part faces the water and hydrophobic part ಎಲ್ಲ ಹೊರಗಡೆ ಹೋಗೋ ತರಾನೇ ಅದು ಅಲೈನ್ ಆಗುತ್ತೆ. That's why you will find a layer of oil on water. Correct? Same way here, ಈ ಕಡೆನೂ ನಿಮ್ಮ ಲಿಪಿಡ್ಸ್ ಇರುತ್ತೆ. These are all the hydrophilic heads. ಇದೆಲ್ಲ ನಿಮ್ಮ hydrophobic tails. ಈ ತರ ಎರಡು ಲೇಯರ್ ಆಫ್ ಲಿಪಿಡ್ಸ್ ಇರೋದಕ್ಕೋಸ್ಕರ we are going to call this as bilipid layer. ಇದಕ್ಕೆ ನಾವು bilipid layer ಅಂತ ಕರೀತೀವಿ. So there are two lipid layers. ನಾ ಹೇಳಿದ್ನಲ್ಲ RBC ಯಾವುದು ಗೊತ್ತಿದೆ ಅಂತ. ಒಂದು RBC ಸರ್ಫೇಸ್ ಏರಿಯಾ ನೀವು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಿದ್ರೆ 2 times the lipids you are get if you extract the lipids from RBC. RBC ಒಳಗಡೆ organelles ಇಲ್ಲ. ಹಂಗಾಗ್ಬಿಟ್ಟು ಎಲ್ಲ ಲಿಪಿಡ್ ಬರೋದೇ only from the cell membrane. ಹಾಗಾಗಿ you will find that it has 2 times the amount of lipids. ಅದಕ್ಕೆ RBC ಬರೋದು. So you have bilipid layer which is here. Okay, ಮಧ್ಯದಲ್ಲಿ ಏನಪ್ಪ ಇರುತ್ತೆ? ಮಧ್ಯದಲ್ಲಿ we will find proteins. As promised, I will use yellow color to show some proteins. ಯಾವ ತರ ಪ್ರೋಟೀನ್ಸ್ ಇರುತ್ತೆ ಅಂತ ಒಂದು ನಿಮಿಷ ನಾನು ಡ್ರಾ ಮಾಡ್ತೀನಿ. ಪ್ರೋಟೀನ್ಸ್ ನ ಡ್ರಾ ಮಾಡಿದ ಮೇಲೆ then I will explain what these proteins, ಯಾಕೆ ಈ ತರ ಪ್ರೋಟೀನ್ಸ್ ಇದೆ ಅಂತ ನಾನು ನಿಮಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. So different types of proteins are going to be present here in this manner. ಈ ಬೇರೆ ಬೇರೆ ತರ ಪ್ರೋಟೀನ್ಸ್ ನಾನು ಡ್ರಾ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. Okay, this is how so many different types of proteins are going to be present here. ಇನ್ನೊಂದು ಪ್ರೋಟೀನ್ ಡ್ರಾ ಎಷ್ಟು ತರದ ಪ್ರೋಟೀನ್ಸ್ ಇವಾಗ ಪ್ರೆಸೆಂಟ್ ಇರುತ್ತೆ. ಪ್ಲಸ್ ಅದು ಕೊಲೆಸ್ಟ್ರಾಲ್ ಡ್ರಾ ಮಾಡ್ಬಿಡೋಣ. ಯಾಕೆ ಬೇಡ? I had told you cholesterol ಇರುತ್ತೆ ಅಂತ. So cholesterol ಡ್ರಾ ಮಾಡ್ಬೋದು. ಓಕೆ, ಲೆಟ್ ಅಸ್ see what we need. ಓಕೆ, ಇವಾಗ ಬೇರೆ ಬೇರೆ ತರದ ಪ್ರೋಟೀನ್ಸ್ ಇದೆ. Proteins which are present can be broadly classified. These proteins we can broadly classify them into 2 categories. Now category proteins are, how do you classify the proteins? Proteins in a cell membrane are classified into 2 categories. One protein is integral protein anta antivi, or some biologists call it as intrinsic protein anta antivi. Integral or intrinsic protein ಅಂದ್ರೆ ಏನು? ಒಂದು ಲಿಪಿಡ್ ಲೇಯರ್ ಅಲ್ಲಿ ಎಂಬೆಡ್ ಆಗಿರುತ್ತೆ. part embedded ಆಗಬಹುದು, partly it might have been embedded, or wholly it might be embedded. But when it is embedded, we call it as an integral protein. ಈಗ ನಾನು ಡ್ರಾಯಿಂಗ್ ಮಾಡಿರೋದಕ್ಕೆ ಇದು ಇಂಟಿಗ್ರಲ್ ಪ್ರೋಟೀನ್. ಇದಕ್ಕೆ ನಾನು 1 ಅಂತ ಹಾಕ್ತೀನಿ. ಅದು ಇಂಟಿಗ್ರಲ್ ಪ್ರೋಟೀನ್. This is an integral protein. Integral protein. This is an integral protein. This one also is an integral protein. These are all integral proteins. They are partly or totally embedded in the bilipid layer. ನಿಮಗೆ ಅದನ್ನ ಹೆಂಗೆ ವಿಜುವಲೈಸ್ ಮಾಡೋದು ಅಂತ ಅರ್ಥ ಆಗದೆ ಇದ್ರೆ ನಾನು ನಿಮಗೆ ಒಂದು ಒಳ್ಳೆ ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ. ಅಂಡ್ ಐ ಆಮ್ ಶ್ಯೂರ್ ಚಿಲ್ಡ್ರನ್ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಇಮ್ಯಾಜಿನ್. ಸರಿನಾ? ಈಗ ಸಿಂಗರ್ ಅಂಡ್ ನಿಕಲ್ಸನ್ ಅವರು ತಮ್ಮ ಫ್ಲೂಯಿಡ್ ಮೊಸೈಕ್ ಮಾಡೆಲ್ ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾ ಇರುವಾಗ ದೇರ್ ವರ್ ಸಮ್ ಪ್ರೆಸ್ ಪೀಪಲ್ ಅಂಡ್ ಕಾಮನ್ ಪೀಪಲ್ ಅವ್ರು ಕೇಳಿದ್ರು, ಏನಪ್ಪಾ ಇದು ಫ್ಲೂಯಿಡ್ ಮೊಸೈಕ್ ಮಾಡೆಲ್ ಅಂತ ಅವಾಗ ಏನ್ ಹೇಳಿದ್ರು ಗೊತ್ತಾ? ಇಮ್ಯಾಜಿನ್ ದೇರ್ ಇಸ್ ಅ ಸೀ, ಒಂದು ಸಮುದ್ರನ ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಆ ಸಮುದ್ರನೇ ನಮ್ಮ ಲಿಪಿಡ್ ಲೇಯರ್ ಅಂತ ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಇನ್ ದ ಸೀ ದೇರ್ ವಿಲ್ ಬಿ many icebergs. Icebergs, I'm sure you have heard. There are many icebergs which are immersed or they are embedded in the ocean. In the same way, you have a sea of lipids, and in that you have these proteins which are embedded. That is how Seeger and Nicholson explain. Now, let me draw it in a brief manner. Let us say, ಇದು ನಿಮ್ಮ ಲಿಪಿಡ್ ಲೇಯರ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಿಸ್ ಮ್ಯಾನರ್ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ನಾವು ಒಂದು 3D ಸ್ಟ್ರಕ್ಚರ್ ಡ್ರಾ ಮಾಡಿದ್ರೆ this is how the lipid layers are present. Okay, let us say that is how they are going to be present here. ಈ ತರ ಲಿಪಿಡ್ಸ್ ಎಲ್ಲ ಒಂದು 3D ನೀವು ಇಮ್ಯಾಜಿನ್ ಮಾಡಿಕೊಂಡ್ರೆ ಎಲ್ಲ ಮಧ್ಯದಲ್ಲಿ ಪ್ರೋಟೀನ್ಸ್ ಅದು ಅದರ ಒಳಗಡೆ ನಿಮಗೆ ಎಂಬೆಡ್ ಆಗಿ ಕೂತಿದೆ. Okay, that is how the proteins are going to be embedded inside. But ಇಲ್ಲಿ ನಾನು 2D draw ಮಾಡಿದ್ದೀನಿ ತಮಗೋಸ್ಕರ. ಅರ್ಥ ಆಯ್ತಾ? Last ಅಲ್ಲಿ sir will give us, Umesh sir will give me the permission. I will share a PowerPoint last ಅಲ್ಲಿ ಒಂದು few slides, 3 slides ತೋರಿಸ್ತೀನಿ. ಅಲ್ಲಿ ನಿಮಗೆ 3D model of the cell membrane ಕಾಣಿಸುತ್ತೆ. ಈಗ ಸದ್ಯಕ್ಕೆ ಬೇಡ. ಸರಿನಾ? So that is how the lipids are arranged, and these are going to be partly embedded proteins or completely embedded proteins are going to be seen, right? ಈಗ proteins, these are all integral proteins. ನಾವು deal ಮಾಡಿರೋದೆಲ್ಲ integral proteins ಆಗುತ್ತೆ. Okay, what about the peripheral proteins? Second type of proteins, ಅಂದ್ರೆ ಎರಡು ತರ proteins ಇರುತ್ತೆ. ಒಂದು partly embedded ಇರುತ್ತೆ, ಇಲ್ಲ completely embedded ಇರುತ್ತೆ. Next type of proteins are the ones which are sitting on the ends of the phospholipids. ನೀವು ಇಲ್ಲಿ ನೋಡಬಹುದು ಅದು ಎಂಬೆಡ್ ಆಗಿಲ್ಲ, ಮೇಲ್ಗಡೆ ಕೂತಿದೆ. This is called as peripheral protein. ಇಲ್ಲಿ ಒಂದು ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ peripheral protein. ಇಲ್ಲಿ ಒಂದು ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ peripheral protein. So some of the proteins will not be embedded. Instead, they are present outside of the bilipid layer. Integral inside the bilipid layer. Okay, now we have all these proteins that are present here. You can see I have drawn some of the proteins in an irregular manner. Some of the proteins are so long and thin. Some of the proteins may be partly embedded. Now the various proteins, even the special protein that you know. Okay, so each protein and exactly. Some of the proteins will have a passage through them. Each proteins channel proteins ಅಂತ ಕರೀತೀವಿ. I'll come to that in the last 5 minutes. ಸರಿನಾ? What is the function ಬಂದಾಗ ನಾನು ನಿಮಗೆ ಹೇಳ್ತೀನಿ. Okay, so proteins ಎಲ್ಲ ನಾನು ಡ್ರಾ ಮಾಡಿಬಿಟ್ಟಿಪ್ಪ. Next ಏನು ಬರುತ್ತೆ? Next is going to be oligosaccharides. ಎಲ್ಲಿರುತ್ತೆ oligosaccharides? As I told you, they are all very small sugars. Small polysaccharides ಅಂತ ಕರೀತೀವಿ oligosaccharides. They will never be present towards the cytosol. They are always present towards the environment. Right? Now, where are these oligosaccharides located? Oligosaccharides can either be attached to the head of a phospholipid. Now, one oligosaccharide, or they may be attached to a protein. When the protein is attached, so they may either be attached to they may be attached to a protein, or they may be attached to a phospholipid head in this manner. So you can see I have drawn some oligosaccharides there. ಒಂದು ಲಿಪಿಡ್ಗೆ ಒಂದು ಶುಗರ್ ಅಟ್ಯಾಚ್ ಆದ ತಕ್ಷಣ the entire molecule, the carbohydrate attached to the lipid is going to be a new molecule. That new molecule is called glycolipid. ಇದು pure phospholipid. ಇದು phospholipid attached to the carbohydrate. This whole structure is called as a glycolipid. I'm sure ನಮ್ಮ ಸ್ಟೂಡೆಂಟ್ಸ್ ನ ಕೇಳಿದ್ರೆ ಖಂಡಿತ ನೀವು ಹೇಳ್ತೀರಾ ಏನು ಇದರ ಹೆಸರು ಏನು ಪ್ರೋಟೀನ್ ಪ್ರೋಟೀನ್ಗೆ ಮತ್ತು carbohydrate has got attached. What is it called as? It is called as glycoprotein, right? This is going to be the structure of your cell membrane. Some of the cell membranes may show the presence of cholesterol also along with them. And apart from that, you will notice that in some cell membranes, some of the cell membranes, you may have another protein called as actin filament, which may be attached to some of the proteins in this manner. So sometimes you may find that actin filaments in the protein. That actin filament may get attached to the proteins towards the inside. ಮತ್ತೆ ಒಂದ್ಸತಿ ಫಂಕ್ಷನ್ ಬರೋಕ್ಕಿಂತ ಮುಂಚೆ and I have not even explained to you ಫ್ಲೂಯಿಡ್ ಅಂತ ಯಾಕಪ್ಪ ಕರೆದ್ರು ಇದೇನಿದು ಲಿಕ್ವಿಡ್ ಮೊಸೈಕ್ ಅಂತ ಯಾಕೆ ಕರೆದ್ರು I still have to come to that, but let us quickly do a recap of the structure. ಆಮೇಲೆ ನಾನು ಫಂಕ್ಷನ್ಸ್ ಆಮೇಲೆ ನೀವಿಂದ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ಓಕೆ, ಸೊ ಒಂದು ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಮಾಡಬೇಕು ಅಂತ ನಮ್ಗೆ ಏನೇನ್ ಬೇಕಪ್ಪ? ಏನಿಲ್ಲ, ಮೂರೇ ಸಾಮಾನು ಬೇಕು. First one is lipids, which may be phospholipids, abundant, and little amount of cholesterol or other sterols. Done. Second component is proteins. So proteins and lipid with proportion can vary from cell to cell in the same person, and definitely between one species and another species, etc. Apart from that, the third component present in least amount is going to be oligosaccharides, which is small type of polysaccharides, or we can just call them in a very common language as sugars. Okay. Now, first to make a cell membrane, you just organize lipids in this manner. We know it on the table tennis balls happy. The killer straws attachment. ಮತ್ತೆ ಒಂದಷ್ಟು ಟೇಬಲ್ ಟೆನ್ನಿಸ್ ಬಾಲ್ಸ್ ತಗೊಂಡು ಸ್ಟ್ರಾ ಅಟ್ಯಾಚ್ ಮಾಡಿ. ಈ ಟೇಬಲ್ ಟೆನ್ನಿಸ್ ಬಾಲ್ಸ್ ವಿತ್ ಸ್ಟ್ರಾ ನ ಹಿಂಗಿಡಿ, ಈ ಕಡೆಯಿಂದ ಹಿಂಗಿಡಿ. Two layers of color, exactly like that you are going to have your bilipid layer. In that bilipid layer, you will embed some or you will arrange some proteins. How do you arrange? If the proteins are partly embedded or completely embedded, integral proteins or intrinsic proteins. If the proteins are not embedded, but they are present somewhere outside the bilipid layer, peripheral proteins, extrinsic proteins, and the peripheral proteins they are going to be. Third component is going to be what? Third component is oligosaccharide. Never present on the cytosol side, but always attached towards the environmental side, and it will be attached to a phospholipid to produce a glycolipid or to a protein to produce a glycoprotein. Idu nimma arrangement. Let us now come to another case in the Nicholson called fluid mosaic model. Fluid and liquid and fluid is called as quasi-fluid nature. So, Singer and Nicholson explained that the word fluid here is trying to mean quasi-fluid nature. What is quasi-fluid nature? It is like a fluid. The fluid word is not used to say it is a liquid. Rather, it is used to say that it is flexible. It has the ability to change. Now, in literature, fluid person describe liquid. I'm trying to say a flexible person. Our ideas now change. That is what is meant by fluid. ಈ ಲಿಪಿಡ್ ಲೇಯರ್ ನ ನೋಡಿದ್ರಲ್ಲ, ಈ ಪ್ರೋಟೀನ್ ಇಲ್ಲಿಂದ ನೀಟಾಗಿ ಮೂವ್ ಆಗ್ಬೋದು. ಈ ಪ್ರೋಟೀನ್ ನಾನು ಇಲ್ಲಿಂದ ಈ ಕಡೆ ಮೂವ್ ಆಗ್ಬೋದು. ಈ ಲಿಪಿಡ್ ನ ನಾನು ಇಲ್ಲಿಂದ ಮೂವ್ ಮಾಡಬಹುದು. So the membrane has the ability to change itself. You can have interchange of the positions. The positions are fixed. These positions can be changed. Yavado one cell, it will decide. Now, these are proteins. It can manufacture within the cell and attach some more proteins. So the fluid membrane is such that it has the ability to change. It is not going to be present in a fixed manner all the time. ಅದಕ್ಕೆ ನಾವು quasi-fluid nature ಅಂತ ಹೇಳ್ತೀವಿ. because you can have the components which can allow lateral movement of the components. ಅದಕ್ಕೆ ನಾವು fluid ಅಂತ ವರ್ಡ್ ಬಳಸ್ತೀವಿ. ಮೊಸೈಕ್ ಅಂತ ಯಾವಪ್ಪ ಹೇಳ್ತೀವಿ? Many of you all have seen mosaic tiles ನೋಡಿದ್ದೀರಾ ಮನೆಯಲ್ಲಿ? Mosaic flooring ಅಂತ. One tile does not resemble another tile. It is very unique. The combination of all those components is very unique. In the same manner, ಇದು ಒಂದು cell pattern ಆಗಿತ್ತು ಅಂದ್ರೆ neighboring cell can have slightly different pattern. ಇದು ಇಲ್ಲೇ ಕೂತಿರ್ಬೇಕು ಅಂತ ಏನೂ ಇಲ್ಲ, ಇದರ ಪಕ್ಕದಲ್ಲಿ ಇದೇ ಇರಬೇಕು ಅಂತ ಏನೂ ಇಲ್ಲ. So the neighboring cell is slightly different. Though they are genetically the same, but there will be slight modification in the way the components are arranged. That's why it is called as mosaic. So fluid mosaic model they are the meaning. Okay, particular membrane, what is it described as? So this is not just structural explanation, it is also functional explanation. In the living world, semi-permeable membrane is very, very rare. ನೀವು ಒಂದು ಮೊಟ್ಟೆಯಲ್ಲಿ ಹೊರಗಡೆ ಒಂದು ಮೆಂಬ್ರೇನ್ ಇರುತ್ತೆ, ಒಂದು ಶೆಲ್ ನ ತೆಗೆದ ತಕ್ಷಣ ಫುಲ್ ಮೆಂಬ್ರೇನ್ ಬರುತ್ತೆ, ನೋಡಿರ್ಬೋದು. That is one of the rare examples of semi-permeable membrane. Semi-permeable membrane will allow only water to pass through and definitely some gases to pass through. But cell membranes are selectively permeable membrane. That is, they allow the movement of many different types of chemicals by actually choosing the entry and exit of the chemicals. That's why cell membrane is described as selectively permeable. Even though amoeba oxygen na ಹೊರಗಡೆ ಬರುತ್ತೆ, carbon dioxide na ಆಚೆ ಹೋಗುತ್ತೆ. Okay, this cell membrane will allow for engulfing, phagocytosis ಅಂತೀವಿ. ಒಂದು ಊಟನ engulf ಮಾಡ್ಕೊಳ್ಳಕ್ಕೆ cell membrane has to grow in that pseudopodia form ಮಾಡಬೇಕಾಗುತ್ತೆ. The cell membrane only can fold inwards to produce endoplasmic reticulum. So ಬೇರೆ ಬೇರೆ ತರ ಈ ತರ ಫಂಕ್ಷನ್ಸ್ ನೆಲ್ಲ ಈ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಮಾಡೋದು ಯಾಕೆ? Because of the fluidity. Let us see what are these components going to do. So when you look at selective permeability, that is, you are allowing molecules to pass through, mainly linked to transport across a cell membrane. One transport is called as passive transport. Passive transport membrane. When the regionally certain molecules are present in higher concentration, in the regionally certain molecules are present in lesser concentration. Higher concentration region and the lower concentration region, when you have substances passing through, that is going to be called as passive transport. Sarina, for example, ಇಲ್ಲಿ ಬಹಳ ಆಕ್ಸಿಜನ್ ಇದೆ, ಅದು ಒಳಗಡೆ ಬರುತ್ತೆ ನೀರಿಗೆ. ಇಲ್ಲಿ ತುಂಬಾ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇದೆ, ಅದು ಆಚೆ ಹೋಗುತ್ತೆ. Okay, that is passive transport. Passive transport ಆಗುವಾಗ, if you don't need the help of any membrane protein, then it is called as simple diffusion ಅಂತ ಹೇಳ್ತೀವಿ. ಯಾವುದೇ ಒಂದು membrane protein ನನಗೆ I don't need its help, I just can pass through the membrane. That is called a simple diffusion. Gases will do simple diffusion. ಇಲ್ಲಿಂದ ಆಕ್ಸಿಜನ್ ಬರೋದು, ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಹೋಗೋದು, ಯಾವುದೇ membrane proteins ಬೇಕಾಗದಾಗುತ್ತೆ, ಅದು simple diffusion ಅಂತ ಹೇಳ್ತೀವಿ. ಸರಿನಾ? Same way, sometimes I need the help of a membrane protein. ಯಾಕಪ್ಪ ಹೆಲ್ಪ್ ಬೇಕು? ಹಂಗೇನೆ ಬರಬಾರ್ದಾ? ಯಾಕೆ ಬೇಕು ಅಂದ್ರೆ, some molecules may be charged particles like ions. Some molecules may be very large. Some molecules may be hydrophilic. ಈ ತರ ಬೇರೆ ಬೇರೆ molecules ಇರುವಾಗ ಸುಮ್ಮನೆ diffuse ಮಾಡಕ್ಕಾಗಲ್ಲ. They need the help of some molecules. For example, other molecules irutte. Now it is beyond the scope of this lecture, but one of the molecules ನಿಮಗೆ ತೋರಿಸ್ತೀನಿ. ಇಲ್ಲಿ ನೀವು ನೋಡಬಹುದು, ನಾನು ಒಂದು ಪ್ರೋಟೀನ್ ಅನ್ನು ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ ಅಂತ. ಆ ಪ್ರೋಟೀನ್ 3D manner ಅಲ್ಲಿ ಹೇಗಾಗುತ್ತೆ ಗೊತ್ತಾ? It will look like a long bead. Bead ನೋಡಿದ್ದೀರಲ್ಲ, ಸರ್ಕ ಹಾಕೋದಿಲ್ಲ bead. It will look like a long bead. ಮಧ್ಯದಲ್ಲಿ ಒಂದು ಟನಲ್ ಇರುತ್ತೆ. Sarina, it will protect that molecule so that it does not, the lipids don't interfere with it, and our molecule is ಅದೇ ತರ you will find ಈ molecule ನಾವು ಮಾಡಿದ್ದಕ್ಕೆ ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ. ಸೊ ಒಂದೊಂದು ಮಾಲಿಕ್ಯೂಲ್ ನೀಟ್ ಪಾಸ್ ಆಗ್ಕೊಂಡು ಬಂದ್ಬಿಡ್ಬೋದು. Such a type of protein is called channel protein. Channel produce ಮಾಡ್ಬೋದು. So that molecules can pass. ಅಂದ್ರೆ ಇಲ್ಲಿ ಸಿಕ್ಕಾಪಟ್ಟೆ ಗ್ಲೂಕೋಸ್ ಇದೆ, ಇಲ್ಲಿ ಕಡಿಮೆ ಗ್ಲೂಕೋಸ್ ಇದೆ. ಯಾಕಂದ್ರೆ ಗ್ಲೂಕೋಸ್ ತಗೋಬೇಕು ಸೆಲ್ ಒಳಗಡೆ ಅಂದರೆ ಆ ಗ್ಲೂಕೋಸ್ ಈ ತರ ಒಂದು ಪ್ರೋಟೀನ್ ಒಳಗಡೆ ಟ್ರಾವೆಲ್ ಆಗಬೇಕು. ಸೋಡಿಯಂ ಅಯಾನ್ಸ್ ಇದೆ. I want sodium ions to travel. They are charged particles. They need a sodium channel. So that kind of diffusion, where you use the help of the membrane proteins, is going to be called as facilitated diffusion. Facilitation and help maadu. Facilitated diffusion and help. So both types are called as passive transport, simple diffusion or facilitated diffusion. Now here transport. Non-transport is called active transport. Active transport andrenu, from the region of lower concentration of a molecule to the region of higher concentration of a molecule, have to send molecules. ಆತರ ಕಳಿಸ್ಬೇಕಾದ್ರೆ ನಾನು ಒಂದು ಮೌಂಟೇನ್ ಇಂದ ಒಂದು ದೊಡ್ಡ ಕಲ್ಲನ್ನ ಕೆಳಗಡೆ ಹಾಕ್ಬೇಕಾದ್ರೆ ಎನರ್ಜಿನೇ ಬೇಡ ಆಗ್ತದೆ. ಸುಮ್ಮನೆ ಮುಂಚಿತ ಅದನ್ನ ನೂಕದ್ರೆ ಸಾಕು ಕೆಳಗಡೆ ಬಂದ್ಬಿಡುತ್ತೆ. ಬಟ್ ನಾನು ಮೌಂಟೇನ್ ಮೇಲೆ ಕಲ್ ತಗೋಬೇಕು ತಗೊಂಡು ಹೋಗ್ಬೇಕಂದ್ರೆ ನನಗೆ ತುಂಬಾ ಎನರ್ಜಿ ಬೇಕಾಗುತ್ತೆ. ಸೊ ಅದೇ ತರ this is active transport andre lower region, lower concentration region and higher concentration region a molecule na transport ಮಾಡಬೇಕು ಅಂದ್ರೆ ನಿಮಗೆ ಎನರ್ಜಿ ಬೇಕು. Energy is ATP. That is called as active transport. ಅದಕ್ಕೂ ನಿಮಗೆ membrane protein help ಬೇಕೇ. Such membrane proteins are called pumps. PUMPS. ನೀವು ನೀರು ಪಂಪ್ ಮಾಡ್ತಾರಾ, ಪಂಪ್ಸ್ ಬೇಕಾಗುತ್ತೆ. ಸೊ ಈ ತರ ಬೇರೆ ಬೇರೆ ತರದ್ದು ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಇರುತ್ತೆ. Sarina. So remember, selectively permeable membrane transport ಮಾಡುತ್ತೆ. Passive ಇರಬಹುದು, simple diffusion facilitated, active ಇರಬಹುದು by the usage of pumps. Okay. So with that, I have come to the end of the class. Umesh sir, if you can just give me permission to share, sir. ಅದೊಂದು permission ನೀವು ಕೊಟ್ಬಿಟ್ರೆ ನಾನು I can just share the PowerPoint ಮಕ್ಕಳು ಅದನ್ನ ಸ್ವಲ್ಪ ನೋಡಬಹುದು ಅವಾಗ. Share ಮಾಡಕ್ಕೆ permission ಕೊಡ್ತೀರಾ sir? If you can just give me permission. Or Siddhegowda sir? Okay, no problem. Sir, you can continue now.
ಮೇಡಮ್ ಒಂದ್ ನಿಮಿಷ ಮೇಡಮ್.
Yes sir, no problem. I think chat ಅಲ್ಲಿ ಏನೋ question ಬಂದಿದೆ ಅನ್ಸುತ್ತೆ. Let me just see. Second year class ಯಾವಾಗ start ಆಗುತ್ತೆ ಅಂತ. Shivkumar, I'm sure your teachers will be in a position to answer that question.
Madam?
Yes, sir.
Yes, ma'am. Students are asking when will be second PU sessions. We will let them know by sending the messages. Is there any interruption, ma'am?
Sir, ನಾನು ಕೇಳಿಸಿಲ್ಲ, sorry. ಹೇಳಿ.
Is there any interruption?
Presentation?
No, ma'am. ಏನಾದ್ರು interruption, any technical difficulties ಅಂತ ಕೇಳಿದೆ ma'am.
ನನಗೆ ಶೇರ್ ಮಾಡಕ್ಕೆ ಆಗ್ತಾ ಇಲ್ಲ ಪ್ರೆಸೆಂಟೇಷನ್ ಇಲ್ಲಿ. ನೀವು
just a minute.
ಸರ್ ನನಗೆ
Just a minute.
I can share.
Yes, ma'am. Degodre, can you please right click on the ma'am's icon and make it pen list? Presenter?
Presenter ಇಲ್ಲಿ ನಂಗೆ ಬರ್ತಾ ಇಲ್ಲ.
ಇಲ್ಲ ನಿಮ್ಗಲ್ಲ ಮೇಡಮ್. I am telling my colleague.
Yes, sorry sir.
So they go to right click on the name of the panelist and make it as presenter. Sorry for the inconvenience, ma'am. Just a
No
second.
problem, sir. That's okay, sir. No issue.
Madam,
Yes,
please try.
yes, I've got the permission. Yes, I'll just share. I'll just share. Yes, students, I think you will be able to see. I will, go to the next slide and you will be able to see. So Illy, you can see Jonathan Singer and Garth Nicholson, Jonathan Seymour Singer and Garth Nicholson. You can see them.
Madam, request you to make
Nicholson
it
is alive with us today. Singer passed away 5 years ago. Nicholson innu nam jothe iddare. And you can see the fluid mosaic model of the cell membrane here. One 3D structure I told you how to see. So ಇಲ್ಲಿ ನೀವು ನೋಡಬಹುದು extracellular fluid, that is environment side, intracellular fluid. ಹೇಗೆ ರೆಡ್ ಕಲರ್ ಇರೋದೆಲ್ಲ ಫಾಸ್ಫೋಲಿಪಿಡ್ಸ್ ಹೆಡ್, ಆಮೇಲೆ ಯೆಲ್ಲೋ ಕಲರ್ ಇರೋದೆಲ್ಲ ನಿಮ್ಮ ಹೈಡ್ರೋಫೋಬಿಕ್ ಚೈನ್ಸ್ ಬರುತ್ತೆ. ಆಮೇಲೆ ಬ್ಲೂ ಕಲರ್ ಇರೋದೆಲ್ಲ ಬೇರೆ ಬೇರೆ ತರ ಪ್ರೋಟೀನ್ಸ್ ಬರುತ್ತೆ. ಪೆರಿಫೆರಲ್ ಪ್ರೋಟೀನ್ಸ್ ಇರ್ಬೋದು, ಇಲ್ಲ ಇಂಟಿಗ್ರಲ್ ಪ್ರೋಟೀನ್ಸ್ ಇರ್ಬೋದು, ನೋಡ್ಬೋದು. ಇದರಲ್ಲಿ ನೀವು ನೋಡ್ಬೋದು ಹೇಗೆ. ಐ ಜಸ್ಟ್ ಮೇಕ್ ಇಟ್ ಆಸ್ ಒಂದು ಸ್ಲೈಡ್ ಶೋ ತರ ಮಾಡ್ತೀನಿ and you will be able to see ಎಷ್ಟು ನೀಟಾಗಿ the molecules are moving through the channel proteins. ನೀವು ಅದನ್ನ ನೀಟಾಗಿ ನೋಡ್ಬೋದು. ಚಾನೆಲ್ ಪ್ರೋಟೀನ್ ಇಂದ ಹೇಗೆ ಮಾಲಿಕ್ಯೂಲ್ಸ್, ಅದು ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ತೋರಿಸಿದ್ದಾರೆ ಇಲ್ಲಿ, ಜಸ್ಟ್ ಆಸ್ಮೋಸಿಸ್ ದೇ ಹ್ಯಾವ್ ಶೋನ್, ಬಟ್ ದಟ್ಸ್ ನಾಟ್ ದಿ ಓನ್ಲಿ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ದಟ್ ಪಾಸ್ ಥ್ರೂ. ಬೇರೆ ಬೇರೆ ತರ ಬಾಕಿ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಪಾಸ್ ಆಗುತ್ತೆ. Okay, that is what you are able to see here, right? Yes, thank you so much, dear students. Thank you for being a wonderful audience. And any questions idre, definitely you can just let me know. Yavade tara questions idru. Over to you, sir.
Thank you very much, ma'am. Such a wonderful presentation. Now I request audience or students, if any questions, they can raise their virtual hand or they can mention it in the chat box. If anyone would like to ask question, we can unmute them. So as of now, there are no questions, ma'am.
No problem,
If
sir.
they feel any doubts or any questions, we will let you know, ma'am.
Definitely, you can forward the questions to me, sir. I can answer and send them across to you. Not a problem. Thank you so much, sir. Thanks for the opportunity.
Thank you very much, ma'am.
Thank you.
Now I request our Siddhegowda sir to give formal vote of thanks. Hello.
ನಮಸ್ತೆ ಮೇಡಮ್.
Namaste, sir.
ಮೇಡಮ್, ಈಗ ನೀವು ಇವತ್ತು ಕೊಟ್ಟಂತ ಒಂದು ಉಪನ್ಯಾಸದ ಮೇಲೆ ಒಂದು ಕ್ವಶನ್ ಕಳಿಸ್ಕೊಡಿ ಮೇಡಮ್.
We request to give some questions on this topic, ma'am.
Sorry, I didn't hear you. Yes, sir.
We request you to give one question for the students because we are planning.
Yes, definitely. Now I will be able to give you subjective questions and objective questions. Subjective questions makle yav tara barutte gotta? Yaru ಈ ಮಾಡೆಲ್ ನ ಡಿಸ್ಕವರ್ ಮಾಡಿದ್ರು ಅಂತ ಚಿಕ್ಕ ಕ್ವೆಶ್ಚನ್ ಇಂದ ಹಿಡಿದು ಸಿಂಗರ್ ಅಂಡ್ ನಿಕಲ್ಸನ್ 1972 ಇಂದ ಹಿಡಿದು ಡ್ರಾ ಅ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಫ್ಲೂಯಿಡ್ ಮೊಸೈಕ್ ಮಾಡೆಲ್ ಬರ್ಬೋದು ನಿಮಗೆ 3 ಮಾರ್ಕ್ಸ್ ಎಲ್ಲ ಬರುತ್ತೆ ಆ ತರ. Or they will say what are the various components and describe the components of the cell membrane. One more question can come. What are the various types of proteins present in a fluid mosaic model of cell membrane? One more question can come. Nimge idralli JEE main type question barutte. For example, they will say that the cell of an animal living in the Arctic region was analyzed. This is going to be the proportion of the proteins and the lipids and other basis mele. Can you tell whether the organism can migrate to a warmer region or not? ಈ ತರ ಕ್ವೆಶ್ಚನ್ಸ್ ನಿಮ್ಗೆ ಒಲಿಂಪಿಯಾಡ್ಸ್ ಆಗ್ಬೋದು, JEE ಮೇನ್ಸ್ ಆಗ್ಬೋದು ಬರುತ್ತೆ. CET ಕ್ವೆಶ್ಚನ್ಸ್ ಬರುತ್ತೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ CET ಸಿಂಪಲ್ ಕ್ವೆಶ್ಚನ್ ಕೇಳ್ಬೋದು. ನಿಮ್ಮ they will say which of these is a correct match. RBC 52% proteins 52% lipids ಈ ತರ ಕ್ವೆಶ್ಚನ್ ಕೇಳಬಹುದು. ನಿಮ್ಮ CET ಬರುತ್ತೆ. NEET ಕ್ವೆಶ್ಚನ್ಸ್ ಬರುತ್ತೆ. For example, they will say why is the fluid mosaic model called as quasi fluid in nature? Very options barutte. It is a liquid, it is present only in aquatic animals, it is made up of only water, or because it has the ability to change itself. Ee tara very questions ಖಂಡಿತ ಬರುತ್ತೆ.
ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
Thank you,
ಇದರಲ್ಲಿ
sir.
ನಾವು ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಮೇಡಮ್ ಕೆಲವು ಪ್ರಶ್ನೆಗಳನ್ನ ಕೊಡ್ತಾ ಇದೀವಿ ಎಲ್ಲಾ ಸೆಷನ್ ಮೇಲೆ ಒಂದೊಂದು. ಆ ತರ ನೀವು ಒಂದು ಪ್ರಶ್ನೆ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಕೊಟ್ರೆ they will write and scan it and send it to us. So best answer will be chosen and they will be given cash prize award. So ಅದಕ್ಕೆ ನೀವು
ಸೊ ಒಂದು ಕ್ವೆಶ್ಚನ್ ಕೊಡ್ತೀನಿ, ಅದು 5 ಮಾರ್ಕ್ ಕ್ವೆಶ್ಚನ್ ಆಗಿರುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ನೀವು
ಡಿಸ್ಕ್ರಿಪ್ಟಿವ್
write it in let us say around 10 sentences ಬರಿಬೇಕಾಗುತ್ತೆ. ಒಂದು ಕ್ವೆಶ್ಚನ್ ಕೊಡ್ತೀನಿ, ಬರ್ದು ಉಮೇಶ್ ಸರ್ಗೆ ಖಂಡಿತ ಕಳಿಸಿ. ಇದನ್ನ ಏನ್ ಕ್ವೆಶ್ಚನ್ ಅಂದ್ರೆ describe the structure of cell membrane. Describe the structure of cell membrane proposed by Singer and Nicholson. Proposed by Singer and Nicholson. Describe the structure of a cell membrane proposed by Singer and Nicholson along with a neat diagram with any 4 labelings. Along with a neat diagram with any 4 labelings. ಆಯ್ತಾ? ಮತ್ತೆ ರಿಪೀಟ್ ಮಾಡ್ತೀನಿ. Describe the structure of the cell membrane proposed by Singer and Nicholson with a neat diagram and any 4 labelings. ಆಯ್ತಾ? ಇದನ್ನ ಬರೆದು ಖಂಡಿತ ಕಳಿಸಿ and all the best to you.
ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್ ಮ್ಯಾಮ್. ಸಿದ್ದೇಗೌಡ್ರೆ ವೋಟ್ ಆಫ್ ಥ್ಯಾಂಕ್ಸ್ ಕೊಡ್ಬೇಕು ಅಂತ ಕೇಳ್ಕೊಂತೀನಿ. ವಿದ್ಯಾರ್ಥಿಗಳು ಈಗ ನೀಡಿರುವಂತ ಪ್ರಶ್ನೆಗೆ ಉತ್ತರನ A4 ಶೀಟ್ ನಲ್ಲಿ ಬರೆದ್ಬಿಟ್ಟು ತಮ್ಮ ವಿವರಗಳನ್ನ ಮೆನ್ಷನ್ ಮಾಡಿಬಿಟ್ಟು ಇದು ಇಮೇಲ್ ID ಮೆನ್ಷನ್ ಮಾಡಿದೀವಿ ksta.webinar@gmail.com ಅಂತ. ಅದಕ್ಕೆ ಸ್ಕ್ಯಾನ್ ಮಾಡಿ ಕಳಿಸ್ಬೇಕು. ಅದನ್ನ ನೀವು ವಾಟ್ಸ್ಆಪ್ ಗ್ರೂಪ್ ಇದೆಯಲ್ಲ ಅದರಲ್ಲೂ ಕಳಿಸಿದ್ರು ನಡೆಯುತ್ತೆ, ಬಟ್ ಇಮೇಲ್ ಮಾತ್ರ ಕಂಪಲ್ಸರಿ ಕಳಿಸಲೇಬೇಕು. ಇಟ್ ಶುಡ್ ಬಿ ಹ್ಯಾಂಡ್ ರಿಟನ್, ನಾಟ್ ಎ ಟೈಪೋಗ್ರಾಫಿಕಲ್ ಇಮೇಲ್ ಓನ್ಲಿ.
ನಮಸ್ತೆ ಮೇಡಮ್.
ನಮಸ್ತೆ
ಮೇಡಮ್, ಇವತ್ತು ತಾವು ಕೊಟ್ಟಂತ ಉಪನ್ಯಾಸ ಭಾರಿ ಸರಳವಾಗಿ ಮತ್ತು ಮಕ್ಕಳಿಗೆ ನೇರವಾಗಿ ತಲುಪುವಂತ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಮೇಲೆ ಕೊಟ್ಟಿರ್ತಕ್ಕಂಥ ಕಾರ್ಯಕ್ರಮ ಭಾರಿ ಚೆನ್ನಾಗಿತ್ತು, ಸೊಗಸಾಗಿತ್ತು. ಮತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್ಗಳು ಎಲ್ಲ ಸೇರಿದ್ದಾರೆ, ಮತ್ತೆ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲೂ ಕೂಡ ಸೇರಿದ್ದಾರೆ. ಸೊ ಹಾಗಾಗಿ ನೀವು ಒಂದು ಕ್ವಶನ್ ಮೇಡಮ್, ಅದು ನನಗೆ ವಾಟ್ಸ್ಆಪ್ ಗ್ರೂಪ್ ಅಲ್ಲಿ ಒಂದು ಕ್ವಶನ್ ಕಳಿಸ್ಕೊಡಿ.
ನಾನು ಕಳಿಸ್ತೀನಿ ಬಿಡಿ. ದೇ ಹ್ಯಾವ್ ಗಿವನ್ ಮೇಡಮ್.
ನಾನು ಟೈಪ್ ಮಾಡಿ ಸಿದ್ದೇಗೌಡ ಸರ್ ಕಳಿಸ್ತೀನಿ ಕ್ವೆಶ್ಚನ್. ನೋ ಪ್ರಾಬ್ಲಮ್.
No need, ma'am. I have taken care of that already. Described it.
Yes,
thank you. Thank you.
ಸೊ ತಾವು ಕೊಟ್ಟಂತ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮ ಭಾರಿ ಚೆನ್ನಾಗಿತ್ತು ಮೇಡಮ್. ಸೊ ಮತ್ತೊಂದು ಅವಕಾಶ ಸಿಕ್ಕಂತ ಸಂದರ್ಭದಲ್ಲಿ ನಾನು ತಮಗೆ ತಿಳಿಸ್ತೇನೆ. ಸೊ ನಮ್ಮ KSTA ಪರವಾಗಿ ಮತ್ತು ನಮ್ಮ KSTA ವೃಂದದ ಪರವಾಗಿ ಮತ್ತೊಮ್ಮೆ ನಿಮಗೆ ವಂದನೆ ಹೇಳ್ತೇನೆ ಮೇಡಮ್. ಥ್ಯಾಂಕ್ ಯು.
Thank you so much.
Thank you very much, ma'am. On behalf of KSTA and on behalf of all the participants, I would like to thank our Chairman sir, Professor S. Ayyappan, CEO Dr. M. Ramesh sir, and all our senior colleagues and Siddegowda and other colleagues who have helped us to take this program continuously and successfully, directly and indirectly. And first and foremost, I would like to thank you immensely for accepting our invitation, ma'am. So on
Thank you
behalf of
so
our
much.
participants and teachers, thank you very much, ma'am. And
Thank
we
you.
will take your help in future lessons and future lecture series also, ma'am. Thank you so much.
100%. I'm
Also,
always
okay, ma'am. Also, I would like to thank our participants who have joined in the WebEx Cisco platform and YouTube platform, along with teachers, students, and science aspirants. Thank you, one and all. See you tomorrow for the next session. Please be ready at 4 o'clock. Thank you, ma'am. Thank you once again.
Thank you. Namaste. Namaste. Thank you.